Well, welcome back, everyone. Today I'm honored to be joined by Dr. Tyler Bland. Thank you so much for joining me.
Great to be here.
Yeah, pleasure having you. Talk to me about NSCLCs. What are they, and how'd your work start with those?
Yes, welcome to science. Lots of acronyms.
Yeah.
So what NSCLC is non-small cell lung cancer. And so lung cancer is a big problem, both here in the United States and then also globally. And lung cancer is actually, it's not the most commonly diagnosed cancer, but it is the one that leads to the most deaths, cancer-related deaths.
Interesting. Okay.
And so my research started to dive into how is lung cancer progressing, how does it respond to therapies, how can we make better therapies, how could we use therapies that we already have, and so forth. And so when we talk about lung cancer, there are kind of two big buckets of different types of lung cancer, that being non-small cell lung cancer and those being small cell lung cancer. So small and then non-small. So when we're looking at those two, non-small cell lung cancer is the majority of what people are diagnosed with. Small cell lung cancer less so. And then you could dig down deeper into the individual ones. There's subsets of non-small cell lung cancer. There's other kind of versions of small cell lung cancer, but those are kind of the biggest ones. And because non-small cell lung cancer is the most common, that's the one that we decided to put most of our efforts into. Most bang for your buck, try and help the most people.
Got it. That makes sense. And what's the point of saying non-small cell as opposed to just large cell? Is there an important distinction there?
There is. So there's small cell and non-small cell, and then in non-small cell, there is a large cell version of it that's actually fairly rare.
Okay.
So you can't just say large cell because that's a small portion of it. Non-small cell is kind of a more general
Medium
term.
to large.
I didn't pick the namings, so it is what it is.
Yeah.
Yep.
What is it about NSCLCs that make them the most deadly as opposed to other kinds of lung cancer cells?
So I wouldn't say it's the most deadly. It is. It's just it leads to the most amount of deaths because that's the most amount of diagnoses. So more people have non-small cell than they do small cell.
Okay,
Small cell is actually typically more aggressive. So you actually end up getting more deaths with that. But because fewer people are diagnosed with it, overall, there's less deaths. What leads to the deaths, though, with non-small cell lung cancer? That's tricky because we have a variety of drugs that work very well. We have a bunch of drugs that work for non-small cell lung cancer and treat patients with varying levels of efficacy. Why do we still get the deaths? And it's largely because of resistance. So you can treat somebody with a drug, that drug works for a certain amount of time, and then the cancer typically becomes resistant and starts to come back, and you have to use a different drug, and then you just recycle that. It becomes resistant to that, and then you use a different drug. Becomes resistant to that, and then you have to use a different drug. And so that's what our research is largely focused on is how does it become resistant to some of these therapies? How can we either block it so that those therapies last longer, or how can we turn the clock back? How can we resensitize cancers that have already become resistant? And so now we can take drugs that previously would not have worked, and now they're back on the table.
Got it. That makes sense. And when did we start figuring out that, hey, these drugs are no longer working? There's something within this cancer cell that is enabling it to develop resistance.
Yeah, so it depends on what type of therapy you're talking about. So there is both intrinsic resistance. That means that the cancer itself already won't respond to it.
Right.
And that's largely when you're looking at targeted therapies. So we have kind of general cytotoxic or drugs that kill just general cells. Those are typically targeting, say, DNA, and almost all cells express DNA, and so it should work. Whereas we have very targeted ones that target certain proteins inside of cancer. And so if the cancer doesn't even express that protein, it's automatically resistant to those therapies.
Got it.
So that's for the targeted ones. For the general ones, how you determine that is largely you treat with it and it doesn't work.
Okay. Got it. And so that's a bit of trial and error there.
Correct.
Is there a way you can determine before therapy starts that this person or these cancer cells are not going to be able to be treated with XYZ drug?
Sure. With the targeted ones, yes. So how diagnosis of lung cancer works is it's typically found with symptomology. You do imaging, you see some sort of mass, you go in, you biopsy it, and then you molecularly profile it. And what that means is that you dig into those cells in that tumor and you figure out what proteins is it expressing. And so if it is expressing a drug target, some of them are like EGFR. This is a particular protein that can be highly expressed in cancer. We have a drug for that. Let's use that drug. If it's not expressing EGFR, you can't use EGFR drugs because it doesn't have it. For the ones that are just broad, general, targeting DNA, there's not a whole lot of things that we can look at to see if it will respond. In general, a faster growing cancer will respond better as opposed to a slower growing cancer. So for instance, small cell lung cancer is very rapidly growing, very aggressive, and typically responds fairly well to therapies, whereas slower growing ones typically don't respond as well. But that's kind of a general bucket. I can't say somebody with non-small cell lung cancer, yes or no, you will respond to these general therapies. It's usually you try it and either it doesn't work or it does.
Got it. And the difference between the small cell seems to move faster, and is it intrinsic in that quick movement that it is then less adaptive or less resistive to therapy, as opposed to the non-small cell, which is moves slower but is a little more fortified?
How it works is that a lot of our drugs are targeting fastly replicating cells, so cells that replicate very quickly.
Okay.
And so the faster that cancer grows, the more the drugs will target them. And because fast-growing aggressive cancers are fast-growing and aggressive, they'll respond better to some of these therapies.
Got it. So it's a bit of they're flying under the radar, the non-small cells.
A little bit, yeah.
A little bit, yeah. What is it? Is that kind of the one of the main reasons why non-small cell is so common, or is there another reason why we see so much less of small cell?
Small cell, it's tricky to say. So small cell is largely induced by smoking. Non-small cell lung cancer can be induced by a wide variety of things. And so it can also be induced by smoking. But because the risk factors for small cell are more narrow, largely developed on cigarette smoking, that's typically why we see less of it. Also, it arises from different cell types, whereas non-small cell can arise from more common cell types in the lung.
Got it. Okay. And there are different types of NSCLCs, correct?
Yes. So going back to the
variants.
beginning,
Yeah.
going back to the beginning, we have small and then non-small, and then inside of non-small, we have other subcategories inside of that.
What identities do they all show? What are the differences between them, and why is it important?
Good. It's largely from what type of cell the cancer arises from.
Okay.
And so inside of non-small, we have squamous cell, we have adenocarcinoma, and then we have that larger giant cell. For squamous cell, these are cells that line the inside of the lungs, and so squamous cells are on your skin, they're elsewhere, and so if the cancer arises from squamous cells, you have squamous cell non-small cell lung cancer.
Got it.
The lung also extrudes a lot of mucus inside of the lung as a protective function. In order to extrude mucus, you have to have glands. And so if the cancer arises from those glands, you have an adenocarcinoma. Adeno just means that it arises from glands. And that's between squamous cell and adenocarcinoma, those are the most common of the non-small cell lung cancers.
What do different therapies look like for each of those?
The therapies are actually fairly similar between the two.
Oh, interesting. Okay.
Yes. And so what typically happens is, like I say, during diagnosis, you go in, you biopsy, you see what it's expressing. And one of the main things you look at when you're looking at what proteins that these cancers express is a molecule called PD-L1. What this is a signal that a lot of cells express, but in particular cancer cells. They can express this PD-L1 receptor, and what this does is this tells the immune system, I am self, don't attack me.
Got it.
And so that's one way that it can evade getting attacked by the immune system. Because typically our immune system's pretty good at removing cancer.
Right.
We have T cells floating around, they find a cancerous cell, it's messed up, and so it kills it. But if it has this little flag, that T cell comes over and it sees it and it goes, oh, yourself, I'm not going to attack you, it moves on. And that cancer is able to grow now and proliferate in wherever it's set up shop. So you look at that first. Is it expressing high levels of this PD-L1? If so, good, because we have a drug for that. So we have a targeted drug. If not, then we have to use some of our broader therapies that target, say, DNA, which a lot of our cells are expressing. And the downside of that is that if you're targeting DNA, which a lot of cells express, yes, you are attacking the cancer, but you're also attacking a lot of other stuff in the body. Yep. And so that's where you get some of these nasty side effects from these.
And that would be chemotherapy, basically.
That would be chemotherapy, correct.
Got it.
Yes.
What are the, from what I understand, it's a genetic alteration in the NSCLCs. Do they start out as something when, can they develop into different genetic variations of small cell lung cancer?
Yes, so they do. They can mutate throughout the progression of the disease, throughout the progression of treatment. They typically, though, do not transfer between the types of lung cancer.
Okay.
So, for instance, you don't get a small cell lung cancer converting into a non-small cell.
Right.
You don't get a squamous cell converting into an adenocarcinoma. You don't see that, but you can see genetic variability inside of the cancer itself. That's indicative of just cancer itself, because what has led to cancer is typically mutations, and those mutations largely break the ability of those cells to fix themselves. So they're messed up, and they become more messed up as they progress. Also, induction of drugs can also lead to messing those cells up. Hopefully we mess them up so much that they die, but not always. So like I said, with our drugs that target DNA, a lot of these drugs, they target DNA to break the DNA.
Right.
If we break enough of the DNA, the cell dies. If we don't, then we lead to mutations, which can lead to more cancer.
Right, and this is like if you don't take all of your antibiotics, it develops a resistance to the antibiotics, and you've got to take stronger antibiotics. Similar kind of thing?
Similar. The problem, though, is that or I should say the nice part about antibiotics is that if you take all of your antibiotics, hopefully you've wiped out all of the bacteria, so it can't build resistance. There's nothing left.
Right.
The problem with cancer is that you can take all of your chemo, but there's two mechanisms of how it can become resistant. Either one, it's intrinsically resistant, which we talked about before.
Yep.
With those targeted therapies. Some cancer cells just don't respond even to our general ones.
Right.
And so we have a whole bunch of different types of cells in that tumor. You have a drug that's targeting, say, 90% of it. You can take all of that and wipe out all 90%, but there's still 10% left.
Right.
So that's the intrinsic factor. There's also the ability of it to mutate to become resistant to those drugs. So not only are there cells in there that won't respond, just the act of treating will convert some into resistant cells. And so treatment will treat somebody, but it can also lead to future treatment resistance.
Got it. And so the cells become anti-fragile, basically, that they more or less become better as their environment becomes more harsh, correct?
Yes. So cancer can do a bunch of weird things. And one thing that it's good at is wherever it has set up shop, it is good at making that home. It's good at telling everything else around it, basically, don't attack me and give me the nutrients that are required for me to grow.
Right.
So
it can lay dormant for several years.
It can. And so one of the treatments, so for instance, it's if you have, this is common in breast cancer, if you have a localized breast tumor, that's great. You can pull that out, but you usually always treat with chemotherapy afterwards. Why is that if you've already taken out the main tumor? The problem there is it's what's called micrometastases. So these are small little cancer cells that have potentially, we don't know, that have potentially gone throughout the body that we can't detect. And so what you do is you pull out the main source, you treat with chemo just in case there's other ones that you can't see, hopefully to wipe those out so that they can't grow and harm the patient.
Got it. What is in the cancer therapy drugs that we're treating these people with, other than chemotherapy? So these more specialized attacking individual strands of protein or DNA. What's in them that allows them to identify the certain strands of DNA and attack it?
So when like I said, when you molecular profile, you're looking for specific targets. If they express those targets, then we use a drug that targets that target. If not, we have to use DNA attackers. And how those DNA attackers work, with lung cancer, there's a bunch. With lung cancer, though, The drug of choice are called platinum drugs. What these drugs are, it's a platinum molecule, literally the metal.
Oh, really?
Yep. And what it does is it has kind of two branches that stick off of it, and it attaches to the DNA. And it can either, because DNA occurs as the double helix, it either links those two strands together, or it links onto a single strand. Now, why does that work? What happens is that as these cells are replicating, they need to replicate their DNA. And how they do that is they take those two strands of DNA, they pull it apart, and they make two more. And then
Right.
they take those four and split and split and split. Now, if you have a drug that's linking those two together, or if you have a drug that's linking just one, what happens is that protein that replicates the DNA, it's speeding along, it hits that drug, and it's not able to pull it apart, and the DNA breaks. And what you're doing
Interesting.
is you're doing that all over inside the DNA. And so hopefully you're just blowing the DNA up so much that that cell dies.
What's the history of how they figured that out?
I don't know. That's a good question.
It's crazy that they would put platinum in your body and it would kill the DNA molecules themselves.
Yes, it is crazy that we put a lot of these drugs in these individuals. And so one of the main things that makes cancer so difficult is that the tumor is you. Like, it's your cells.
Mm-hmm.
And so the thing is, how do I kill the tumor you before I kill the you?
Yeah.
And that's really why our cancer therapies are so nasty.
Yeah.
Is that it's linking DNA, it's causing DNA breaks, it's doing it in the cancer, yes, but it's doing it everywhere else.
Right.
And so I think in the future, as we get more and more of these targeted therapies, these kind of general chemotherapies that target a bunch of stuff, we're going to hopefully start phasing those out because the side effects are so nasty.
Yeah, it blow everything up, and that doesn't always. There's civilians in there too.
Correct. Yep.
Yeah.
There are so many civilian casualties
Yeah.
in cancer therapy.
Yeah. Talking about that anti-fragility, that ability for the NSCLCs to develop resistance over the course of time because of treatment. They kind of receive the treatment and then develop some sort of combat against that.
Mm-hmm.
Is there anything we can do that inhibits that so that it only targets in other words, when it shows up, it only hits the things that it knows it can kill, as opposed to hitting everything and maybe giving something the genetic code it needs to mutate and grow in advance?
There's a couple different things that you can try to do. How typically cancer resistance works is that, say the cancer is feeding off of protein X. So you target protein X. Cancer is very good at going, fine, whatever, I'm going to use protein Y. And so it flops over to that. Or it uses something downstream of protein X that bypasses the drug that you're using. So you could do a couple different things. One way that people are treating is that some of these resistance mechanisms occur commonly. So if it drives off of protein X and then typically it switches over protein Y, what you can do is treat with a drug that targets X and then another drug that targets Y.
Right.
So that it has to go through multiple steps to try to get through resistance. You can do that. What my lab is trying to do is not how can we bypass that, but how can we reverse it? So how can we, if it's using protein X and then it becomes resistant and goes back to protein or goes to protein Y, how can we move it back, force it back to be using protein X again so that that original drug continues to work?
What have you found thus far?
We found that it depends on the drug you're using, but sometimes this will work. And so one of the drugs that we are looking at it's called a taxane drug. This is a drug that blocks what are called microtubules. And so when a cell replicates, what happens is that the cell grows, it divides its DNA, it replicates its DNA, and then all that DNA lines up in the middle, and then it sends out what are called microtubules. You can think of it as kind of like scaffolding on the side of a building.
Okay.
But what it does is it starts, you have microtubules on both sides, grows out, it grabs that DNA, pulls it to the opposite sides of the cell so that the cell can now split into two daughter cells. Okay? So
Right.
it's essential that those microtubules grow out, grab and pull it. We're looking at a drug that blocks the ability of those microtubules to grow out. Okay? So
Got
they
it.
get stuck, they can't grow, and then the cell dies because it's not able to replicate.
Right.
This is typically used, at least in lung cancer, as a last line. So you've used some of your targeted, you use some of your platinum base, it's become resistant to all of these. This is kind of last line, let's try this. Typically it becomes resistant. And then you have the last option, which is usually the patient passes away. What we want to do is extend the life frame of that drug. So you use that last Hail Mary, but it lasts much, much longer because it doesn't become resistant. So we've blocked that resistance. Not only that, in patients that have become resistant and their cancer is spreading, we can again turn the clock back and use that last Hail Mary again so that it lasts longer.
Now, these microtubules, it sounds like when you're blocking those, you're just blocking it from advancing. How are you then turning back the clock and reversing the proteins from Y to X?
Good. So what we're looking at is we have cancer cells, human cancer cells from lung cancer patients, and we look at patients that respond well to this therapy, and we look at patients that are resistant to it. And we try to see what's the change. What's going on that the cell is now like, I don't care about that drug, I'm going to grow anyways. And one receptor that we have found is called muscarinic receptors. These are receptors found throughout the body. In the lungs, and they're normally expressed in the lungs. But what we find is that when it becomes resistant, there's even more of this receptor. And so we thought, maybe this is what's it. Maybe this is what's causing it to become resistant. And so what we did is we have, again, two cells. We have cells that are sensitive to the chemotherapy. We add the chemotherapy agent, cells die, which makes sense because they're sensitive. Then we have a dish that they're resistant. We add the chemotherapy, they don't die, they keep growing. That was to be expected. But we have high levels of these muscarinic receptors. So we have drugs that block this, luckily. We have drugs that have been used for decades in the clinic. One is used to treat IBS, irritable bowel syndrome. One is used to treat overactive bladder. What's nice about that is that they've been in the clinic forever, very safe. We don't get these nasty side effects that typical chemo does. So we're like, heck, let's try it. So we add this drug to our resistant cells. Nothing happens. We were like, darn it. Like, I thought this was going to work. But then what we did is we added the drug, the IBS drug or the overactive bladder drug, and then we added the chemotherapy drug on top of it, and they all died. And so what's going on is that these really well-tolerated drugs aren't killing the cancer, but it's sensitizing it. It's turning the clock back so that you can use the chemotherapy, and then it'll wipe them out. Now what's even cooler is that we did the same thing to the sensitive cells. So we added chemotherapy, they die. That makes sense. We added the muscarinic drug. Didn't do anything. That's what we saw before. We added the chemotherapy drug, the combination, the anti-muscarinic and the chemotherapy drug, and the chemotherapy worked better. What's nice about that is like what I've been talking about so far is a lot of these drugs that target everything in the body, you get these nasty side effects. So what if we could treat with both drugs, and the one that has the nasty side effect, we can use just a little bit of it.
Use much less.
Exactly.
And get the same effect.
Exactly. So in patients that will respond to the drug, you can use less of it. In patients that are resistant, you can still use it because we can resensitize those cancer cells to that drug.
Yeah, that's wild. What's happening on a molecular level when you introduce that first drug right before the chemotherapy?
Great question. We are currently researching that. And so there's big signaling pathways inside of the cell that are downstream of these receptors. We're currently trying to tease out what exactly is going on. But that work is ongoing. I can't tell you that yet.
Yeah, fair enough. Is the goal to eventually escape chemotherapy altogether? Or is this kind of a curse we're going to have to deal with as far as we know for the rest of time?
Unfortunately, it's probably the latter. Like I said, we're using a drug that doesn't kill cancer cells but resensitizes them or sensitizes them in the original state. So you unfortunately need that chemotherapy on board. I think, like I mentioned earlier, what's going to happen is that we're going to start phasing out some of these drugs that are really nasty side effects as we're getting more targeted ones. And then the more targets we find and the more drugs we are developing in order to target those, I think chemotherapy will get even better. But we have to get to that stage first.
Right. Have you experimented with adding that anti-musk something?
Anti-muscarinic. So it's a muscarinic receptor, against it.
Anti-muscarinic receptive drug, and then adding one of these personalized, very specific custom therapies? Or are you just using broad chemo after that?
Good. That's a great question. For most of our stuff, we're using broad chemo. We have looked at one particular targeted therapy. I mentioned this one earlier, EGFR. This is a receptor that can be highly expressed in some cancers, lung cancer being one. We have looked at cells that respond to EGFR inhibitors, cells that are resistant to it, and then we did the same thing, combination therapy. It looks like our combination therapy might help a little bit, but not as much as some of the bigger cytotoxic ones that we've looked at.
Interesting. Why is that? Because surely once you're opening up the sensitivity, the drugs would just become more effective, right?
It depends on what the drug is doing. And so if the drug is blocking microtubules, it looks like muscarinic receptors are involved in that resistance pathway.
Got it.
If the cell is being fed by EGFR, which is over here, it's potential that there's an entirely different resistance pathway that's involved in that. Maybe has nothing to do with muscarinic receptors.
Right. Is there any? Has there been any field of study that has looked at? Well, let me ask it this way: what is the purpose of muscarinic receptors in the first place?
Oh, good. Yes. So muscarinic receptors are expressed in healthy tissue, and it's involved in the parasympathetic response in the lungs. So your rest and digest. Parasympathetic is rest and digest. Sympathetic is fight or flight. So in rest and digest, you can calm down. You can clear stuff out of the lungs because you're not having to, say, run away from a bear. And so in healthy lung tissue, what it does is it promotes secretions. So secretions of mucus into the lungs so that you can clear any junk out that you've been breathing. On the reverse side, in the sympathetic system, you want to open those lungs up so that you can, say, run away from the bear. So there are normal responses, I should say there are normal functions of these receptors in healthy tissue. What happens in cancer a lot of times is that some of these healthy pathways get hijacked, either overrun if it's like pro-growth or if it's anti-growth, completely shut down. And that's one way that the cancer can grow so rapidly, can get away from the immune system, can increase its environment around it to either increase blood supply or increase the necessary molecules in order for it to grow. And so cancer uses a lot of the normal stuff. It's just that it gets hijacked and overrun.
Got it. So these muscarinic receptors are more or less a neutral party. They're just doing their job. Unfortunately, the cancer is asking them to do their job. Is that correct?
Most are. Most things in cancer are just doing their job. It's just they're either doing it too much or they're doing it inappropriately.
Got it. Okay. So is there what's the earliest stage you can kind of pull these muscarinic receptors back without damaging the healthy tissue? What's the kind of threshold there?
You can block them at any point. And so, like I said, the drugs that we're using that are blocking those receptors, they are irritable bowel syndrome drug, and they are an overactive bladder drug. So people with irritable bowel syndrome or overactive bladder can take these drugs to treat their disorders, and it's not going to mess up with their lungs. It'll treat that specific disorder.
Got it, yeah.
And so, like I said, the side effect profile of these are very well tolerated.
Yeah. Are there situations that are necessary for the cancer to develop this resistance, not inherently, but as it receives treatment, or is there have we not figured out what causes them to initiate this resistance?
What causes them to initiate resistance is typically the therapy itself. And so as cancer is using, like I said, a certain pathway, protein X, it will continue to use protein X as long as it can. And then once you block it with drug, you now force it to mutate to start using protein Y. Correct.
Right.
So we are the ones who cause resistance. Unfortunately, you can't say, okay, I just won't treat you so then you won't get resistant. Then you're not treating the cancer and cancer progresses anyways.
Cancer wins. Yeah.
Correct. Yep.
Is there anything we can... are we able to identify that the cancer is likely to jump to protein Y before it has?
That's a good question. As I mentioned earlier, the progression, sometimes the progression is predictable. Like it'll go from X to Y. And so in those cases, if we have drugs, we can treat X and Y. In other cases, we have no idea what the progression is, and we don't know what. So it's using protein X, and then we block it, and then all of a sudden it's using protein ? We don't know what it's doing, but it's still being able to be resistant to those therapies. And so a lot of research is going into resistance to each of the different types of drugs. How is it doing that so that we can block it? For ours, we're looking at those microtubule poisons. Like when it becomes resistant to microtubules growing out and it still does it, what's the mechanism? That applies to those microtubule poisons, but it might not apply to anything else. So it's not always predictable.
Got it. And yeah. How do you once a person has you've treated them and you've seen that it's been unsuccessful or not entirely successful, what is the protocol for determining what happened and what went wrong?
There's basically treatment algorithms on that. You start with this drug. If that doesn't work, you move to this drug. If that doesn't work, you move to this drug. That's in general. Again, if you have targeted things, so say for instance, somebody they have cancer, you molecular profile it, and it's not expressing EGFR. So you don't use an EGFR targeting drug. Use something else. As the cancer progresses, you will continue to biopsy and see what's going on. So if you biopsy and now it's expressing EGFR, sweet, now I have a new target that I can use.
Right.
So it all depends on the progression of the cancer. How is it mutating? What is it starting to express? And what is it no longer expressing?
Got it. Okay. And there's no way of determining before the cancer even sets in that this person is either genetically or epigenetically has inherently resistant or anti-fragile cells.
It depends on what proteins is expressing. So is it expressing PD-L1, that self signal? So if it's expressing that self signal, it's resistant to the immune system, the patient's immune system.
Right.
But that gives us a drug target that we can try to block it.
Yeah.
If it's not expressing a target, then it's resistant to our drugs, at least our targeted drugs. It doesn't mean that it is non-responsive to other ones or that it's more aggressive or not. It just depends on what it's expressing.
Yeah. I guess what I'm trying to get at is, are you able to profile a person as this person based off their cells before they go cancerous, has cells that are more likely to resist or grow combat mechanisms to the therapy before they get cancer? Is there a profile that we can attach them to, or is it kind of they get cancer and then they resist, but there's not really a determiner before they get cancer or during the original cancer treatment or identification that this person's going to be?
That's a good question. And so when we say a patient has cancer or doesn't have cancer, there is kind of like pre-cancer. So in order for a cell to become cancerous, it has to have 6 to 8 mutations in order for it to continue to grow, evade the immune system,
right?
No longer listen to the signal to stop growing. So it has to get a bunch of mutations. So you can get a spectrum inside of that where it has 1 or 2 mutations, maybe 3 mutations, maybe 4 mutations, which is not cancer, but it is growing abnormally.
Right.
In some instances, you can predict, will this progress to cancer? Typically, though, you don't treat with chemotherapeutics in advance of that.
Before.
Correct. So if somebody doesn't have cancer, you're not going to treat with a cancer therapy.
Right.
That's usually saved for when they actually have cancer. There are, though, some treatments you can do. So, for instance, in the colon, you can get colon polyps, which is outgrowth of cells into the colon itself. Those are abnormal growths, so they typically have some sort of mutation that's causing them to grow, but it's maybe not necessarily cancer yet. Treatment of those is you snip them out. You don't allow them to keep growing and keep building up mutations in order to eventually become cancer.
Right.
But like I said, you typically don't treat with chemo at those early stages.
In the pre-cancer.
Correct.
Is there, are you able to identify which cells are going to become resistant before they become resistant?
Not unless in that precancerous stage, one of the mutations is a mutation that we see later in chemotherapy resistance.
Okay.
And that's all going to depend on what proteins it's expressing again and what drugs we have.
Right. Yeah. Are there any non-cancer, as you're looking at treating from a current science perspective, before we've introduced what you're working on?
Mm-hmm.
Are there any non-cancer drugs that you've seen efficacy in besides this bladder drug that similar to wasp nests or wasp stings and breast cancer? You see efficacy in treating cancer in a somewhat roundabout manner.
Yeah, there's actually a lot of research going into either natural compounds or repurposed drugs in order to treat cancer. The downside of that is that it's not typically patentable, and if it's not patentable, pharmaceutical companies don't care about it.
Yeah.
And so that's why it lands in the plates of scientists in order to go, okay, I don't care about the money that this is going to draw. What I care about is the money that it's going to save the patients. That, okay, I could take a chemotherapy drug that costs thousands of dollars, or I could take maybe aspirin. This is totally hypothetical.
Right.
Again, there's no money, though, in selling aspirin to chemo patients or to cancer patients. So there is a lot of research. Off the top of my mind, I can't think, though, of particular drugs that are being used, but there's a lot of research out there that is doing that.
Got it. And is that the same for repurposed drugs? Because surely the drugs that are already patented as, treating one thing, but could also be used to treat this other form of cancer.
Mm-hmm.
Pharmaceutical companies, it sounds like, would have more interest in those, given that they already have a patent. Is that correct?
It depends. So for when we say repurposing, what we're doing is we're looking at other areas of the clinic outside of oncology, outside of cancer treatment, and going, what drugs do you guys use that we could use in order to treat cancer? That's repurposing.
Right.
Now, if a drug is still on patent, then a pharmaceutical company would probably be interested in repurposing. So, for instance, we have COPD drugs in patients with COPD that target muscarinic receptors. Some of those are still on patent. So if I could go to a pharmaceutical company and go, okay, your patient population is COPD patients, I'm going to expand your patient population to COPD patients and one of the most highly diagnosed cancers in America. They would be interested in that.
Right.
If I go to a pharmaceutical company and I go, here's a generic med that's super cheap, that's used in overactive bladder, you can now use it in cancer patients, they're going to go, I don't care. Like, I'm not making any money off of that anyways.
That seems like a perverse incentive.
Yes. Unfortunately, that is the way it is, though.
Is there any steps being taken to fix it?
Probably outside of my pay grade.
Yeah.
What I do is that luckily I'm in the lab, and so I can research whatever I want, regardless of how much money it might make a pharmaceutical company.
Yeah. Yeah, because that does not seem like the best way to go. Like, surely you want to help people, not just make a whole bunch of money.
Mm-hmm.
But I guess here we are. Are we able to manufacture anti-fragile drugs in the similar way to the anti-fragileness of these cancer cells? That the drugs come in, they knock off all of the cancer cells that are using protein X, and then they notice that there's no more, that there's more things there, but they're not using protein X, and so they move to protein Y.
Mm-hmm. I'm sorry, the question is, can we make these drugs?
Are we able to manufacture those kinds of drugs? Yeah.
Are we able to manufacture them? I mean, the answer is yes. The question is, do we have a drug that we know targets protein Y, for instance? So it's not the problem of can we make it, it's the problem of can we discover it. So can we discover a drug that targets that particular protein? And then once we do, it's usually pretty easy to make it.
Right.
It also depends on what type of drug it is. So we can put drugs into different buckets. Some of them are called small molecules. These are chemicals. Chemicals are typically pretty easy to make. We can make them in a beaker, for instance. vastly simplified. Those are typically, I'm going to say, cheaper to make, not necessarily cheaper to buy.
Right.
And then there are drugs that target, say, our PD-L1. These are antibodies. Antibodies are proteins, way more complex, much harder to make. And so those are more expensive typically to make, and thus more expensive on the patient end. That's my long-winded answer of going, yes, we can make these drugs, but we have to find them first.
So our problem is not identifying the cancer cells and what they're mutating to. It's rather identifying something that can treat them given their current mutation. Is that correct?
No, it is twofold. And so when they mutate, what are those targets? A lot of research is going on to, like, what is it mutating into? What is it now feeding on? Like I said, we see it's using protein X, and then it could swap over to protein Y. A lot of times it's using protein X, and like I said earlier, it's swapping over to protein question mark. We don't know. We don't know what it is.
Right.
So we need to not only find the target, but then find a drug that targets that target. And hopefully that drug is specific in targeting that one, because going back to what we talked about earlier is that I'm targeting you. Like, the cancer is you. It's a messed up form of you, but it's you. So unfortunately, your cancer cells express protein X. A lot of your other cells also express protein X. How can we find a target not only that the cancer is feeding off of, but hopefully the cancer itself has more of or is dependent more on than the rest of your cells? Because if we block it, we want to just block the cancer. That's ideal.
That's the goal.
Yep. In reality, that doesn't happen because we block cancer and then we block everything else. But if cancer really, really, really needs protein Y, let's block that, and the rest of your cells like, nah, I can use something else. That's a great target. Let's target that and kill cancer and not you.
Yeah. What's the current status on T-cell therapy when it comes to dealing with NSCLCs?
Great question. So T-cell therapies, there's some super sexy therapies out there for T-cells. It's called CAR T therapy, chimeric antigen receptor T-cell therapy. In general, what it is we can pull out somebody's T-cells, genetically modify them to kill cancer cells, and inject it back into the patient. And then those cells are able to go find the cancer and kill it. They're really good at treating liquid tumors, so blood cancers. They're not really good, at least right now, at treating solid tumors. And our non-small cell lung cancers are solid tumors, correct. It's very tricky for those T cells that you infuse back into the patient to get into the tumor to kill it all. But also, as I mentioned earlier, wherever the cancer is setting up shop, it likes to set up an environment that is pro-cancer and typically anti-immune system. And so if it's setting up that environment, those T cells come in, they can't penetrate, and the ones on the surface they can't get because the cancer's going, no, not here. Move along.
Interesting. Now, let's start at the beginning. How do they extract T cells from someone?
It's just a blood draw. So you have T cells going through your system all the time.
Yeah.
You can pull somebody's blood and you can extract just T cells.
How do you get the T cells from the once you've blood drawn?
I don't know the particular mechanism, but
Okay.
but we have protocols of you can spin stuff down. You can have certain things that bind to T cells to pull them out. There's very different ways that you can do that. But you can isolate somebody's T cells.
Got it.
And then what you can do is, how do you genetically engineer them? I assume that was going to be your next question.
Yes, more or
Yes.
less.
I'll take a quick step back. When I talked about PD-L1 expression, so if the cancer is expressing PD-L1, that's that self signal to tell T cells like, I'm self, don't kill me. We have drugs for that, and they're called antibodies. Same exact thing that your body produces when you have an infection. So say you have an infection with some bacteria, your body is going to produce antibodies, and these antibodies, they look kind of like Ys. They bind to a specific thing. They're going to bind to that specific bacteria you were infected with. They're going to bind to a specific virus that you were infected with. We can engineer them to bind to PD-L1. So it's going to bind to PD-L1, and it's going to cover that up so the T cell can't see it, and if it's not seeing that self signal, it can then attack it. Now we can harness that. If we can find something that's particularly expressed on a cancer cell, say protein X, there's a lot of protein X sticking out of a cancer cell. we can make an antibody against it. But we don't want to block it. What we want to do is give it to a T cell, attach it to the mechanism to turn that T cell on, so that I infuse the T cell, it goes around, and then once that antibody binds to the cancer cell, it goes, oh, I'm supposed to kill this. And so you're specifically targeting, you're making the T cell a heat-sinking missile that goes in and binds and attacks only something that is expressing protein X. Super cool stuff.
How does it deal with the once the cancer cell says, all right, I've got protein X, they killed all of that, now I'm going to move to protein Y. Do you have to re-engineer the T cells at that point?
Yes. All of the T cells that you have made are now no longer useful. Yeah. They have removed the target, and so the T cell doesn't know what to attack. Not great.
Got it. Yeah. What have been the best shots at creating some sort of similar T cell therapy for solid tumors?
It's the same. It's the same idea. Same idea for hematologic, so blood cancers, as solid tumors. It's just that it works better in liquid tumors and blood cancers, and not in solid. They're doing a variety of different things to try to increase its ability to penetrate into the tumor, its ability to recognize these certain molecules to attack it. There's also some people that are looking at macrophages. This is a different type of immune cell outside of T cells. These are the ones that you think of. They're big. They're going to gobble stuff up, so they eat up junk. They eat up bacteria and stuff like that. What's cool about these is that, I shouldn't say it's cool, is that there are macrophages that eat up bad stuff, and there's macrophages that help with healing and like growing. The cancer hijacks the macrophages to help it grow, to help it heal. And so you have a lot of macrophages in that tumor that are like, yeah, I'll help you out. But there's versions that attack it. And so there's a lot of research going into macrophages are already in that tumor. How could we flip the switch from I'll help you out to I'll eat you? And so that's another branch that we're trying to activate the immune system is that maybe we can't get the T cells to get in. But what's already there? Macrophages are. How can we utilize those?
Yeah. Have they had any luck with that?
Nothing that is FDA approved as of now.
Okay.
But at least in a dish, there's some promise there.
Okay. Well, that's good to know. How do you and your team go about studying these NSCLCs? You mentioned a little bit about the chemotherapy and adding these, I've forgotten the word again, but something long and starting with
Anti-muscarinics.
Muscarinics. What else are you and your team doing as you're seeking to discover more about these NSCLCs?
Mm-hmm. So what we're doing is, as I mentioned, maybe it's using protein X, and then it becomes resistant, and it converts to using protein ? We're trying to figure out what's that question mark. And so how we do that is we have cells that are sensitive to chemotherapy, and then we just keep hitting them with chemotherapy. We give them more and more and more and more until they become resistant. So we're trying to mimic what happens in an individual, where they originally respond, but as we treat, the cancer mutates into a resistant form. So we're trying to mimic that in a dish. So if we continue to treat, it now becomes resistant. Now what we're doing is we're able to pull out not only the DNA that those cells have, but the RNA, which codes for the proteins. I won't get into depth in that. But basically, what is going on inside of the cell? What is that question mark that they're now expressing a lot of, or maybe they're expressing a little of, and can we target that? And so we're starting off with muscarinic receptors. We found that there's high levels of muscarinic receptors. Maybe that is our question mark. Maybe that's what it's flipping over to, and that is causing the resistance. And when we use a drug to block it, it looks like we revert it. So that might be our question mark. I think, though, that there are a lot of other question marks there.
Right.
And when we get this data back, there are hundreds of different things that are either there's too much of it there or there's too little of it there. And so going through each one of those takes a lot of time. And how we started with muscarinics is, one, there was a lot of it. That was one of our biggest hits. But two, there's already a drug to block it, and already a drug used in people.
Got it.
The downside of developing a new drug is that you have to go through all of the clinical trials, phase 1, 2, and 3.
Yep.
And most of the time when the drugs fail, it's for two reasons. Either one, the drug didn't work, or two, it was too toxic and people couldn't take it. Because we're using drugs that have been used in the clinic for decades, it's safe. We already know it blocks it. We're just trying to see, hey, can we use it in cancer now?
Yeah.
Whereas if we found a brand new protein, we have to develop a new molecule that's blocking it. That takes some time, not only just to find the molecule, but then is this safe in humans? Does it actually work in a real human and not just in a dish? It takes much, much longer to do that.
Yeah. So repurposing the drugs is much easier.
Quicker, cheaper.
Yeah. Where are you getting these cancer cells from that you're experimenting on?
They are commercially available cancer cell lines. And so they have, there were patients originally that had lung cancer. They pulled those out. And because cancer is so good at growing basically forever, you can just keep growing those cancer lines for forever. And so there are companies out there that have multiple cancer cell lines that have come from multiple different patients that you can just keep growing and keep experimenting with. And so those are the ones we are using.
So you can just buy cancer.
You can buy cancer.
That's kind of a little terrifying. But
yes.
But I guess the cancer only works in the person it was originally developed in. Right?
Correct.
So
if I was to inject it into myself, for instance, very, very unlikely that it would grow. One, because my immune system not only would recognize it as cancer, but it would recognize it as foreign.
yeah, it's something else.
Correct. And so if you have patients that need say a kidney transplant, I take your kidney and I put it into me. I can't just do that and my immune system will be cool with that. I have to tell my immune system, no, don't attack anything. Like, chill out. As long as I'm not doing that to myself, if I inject these cancer cells into me, I should be fine. I'm not recommending anybody do that.
Don't try it. Yeah.
I'm not saying do that. I'm not saying I would do that. I'm just saying it's not as scary as people typically think it is.
Out of curiosity on the kidney thing, how do you tell your cells to chill out and not kill you?
We have a lot of drugs that are very good at suppressing the immune system.
Okay.
So in cancer therapy, we want to activate the immune system. In transplant, we want to inhibit the immune system. So there's kind of both edges of the coin, depending on what you want to do.
And then once it's in there, it's kind of okay with it, or?
Those patients typically have to be on those drugs for life.
Oh, okay. Got it.
Yeah. So what you really don't want is to get a kidney transplant and have cancer at the same time, because I don't know what to treat you with.
You're kind of doomed on that front.
Yep.
That would be a bummer. As far as the chemotherapy, you give it enough of it, it will develop resistance to chemotherapy. Are there any drugs we have found that are 100% effective at just killing everything and have no downside where the cancer cells can develop resistance to them?
No. There's no such thing as 100% effectiveness with no side effects. Like that would.
Well, not with no side effects, but like with without the specific side effect of the cancer developing resistance.
It depends on the cancer type, it depends on the drug, and it depends on how progressed the cancer is. Typically, when we're saying something is curative, when we are curing cancer, you are surgically removing that cancer. If you're at the point where you're using drugs systemically, like injecting it into a person, typically not curative. Typically, you're just buying time.
Interesting. And so there's always been this study to cure cancer.
Mm-hmm.
Is there ever going to be a point where you think we will get there?
Some you can. Some cancers you can't. Like I said, some you can cut out.
Yeah.
Let's see. Testicular cancer. Actually, that's not a bad one to get because you do treat with a curative intent. So I do give systemic drugs and you can cure people with testicular cancer. For the wide variety, though, of cancers, most of it, once it's spread, it's not curative.
Yeah.
Are we trying to develop drugs that end up curing cancer? Of course. Of course. But we're not there yet.
Yeah. When you and your team are doing research on these NSCLCs, how do you account for the wide variety of genetics across all humans?
Great question. So for all of our research, we use at minimum 2 different types of cells. So cancer from you and cancer from you that we can buy. That's at minimum. What you really want to do is use 2, 3, 4 different types to see, is the same mechanism of resistance, is it a general thing? Like, would your cancer respond and move from X to Y exactly as my cancer would respond and move from X to Y? And I have to use multiple cell lines in order to do that. If I just use one, I'm just looking at, does this drug work in you? If it does, cool. Will it also work in me? I don't know.
Right.
Because I have an N of one. I have a single patient. So what we're trying to do is have multiple patients in a dish to see if it works.
Got it. And how many buckets would you say you've come across of these are the different kinds of reactions, the different kinds? Is it completely different for every individual person, or are there kind of broad categories that people can fit into?
For like resistance?
Yeah, for resistance and for just general the kinds of cancer cells you see.
We how do I want to answer this? We are getting less and less into what are the big buckets and more into the personalized medicine. Like, what exactly is your cancer expressing? What exactly is your tumor doing? Because the better we can understand exactly what yours is doing, the better we can treat. The argument is, okay, I make a drug, is that going to work for everybody? Probably not. But do I want to make a drug that works a little bit for everybody? Or do I want to make a drug that works like a champ for a few people and then not for others? Let's find another drug for those other people.
Yeah.
I'd rather have a drug that cures cancer in 10% of the people than
and have 10 of those drugs.
Yeah, and then kind of treat everybody's cancer.
Yeah.
That's what we're getting into in personalized medicine.
Right. Yeah. How are you and your team? Are you using artificial intelligence at all? I know in breast cancer they've recently found that AI is much better at detecting breast cancer even before, like in the pre-cancer stage, as it's in the third or fourth mutation. Are you and your team using artificial intelligence at all in your study of NSCLCs?
We are not, but I know there is a lot of research out there. The benefit of using AI, especially in like my type of research, is, as I mentioned earlier, when we get these cells that become resistant, we pull out their RNA to see what proteins there are, and there can be hundreds of different things that are going wrong. It's difficult for a human to go through these large swaths of data and understand all the connections, which one's doing what. What's nice about AI is that it can ingest so much data, synthesize it down, and then spit out, this is the most likely candidate. And there is a lot of research going on out there that's now using that, not only to determine what are the targets, but on the chemistry side, how can we make new molecules that will specifically bind to that target?
Right.
And I've seen some new recent papers coming out that's showing that AI is a lot better at it than we currently are.
Yeah.
And so my hope is that not only our number of drugs will start increasing that has availability in order to patients, but the specificity of the drugs will increase, and that it will only bind to protein X, which is really expressed in the cancer and not anywhere else.
Right. Is the work you're doing, have you seen it have any effects on cancer research more broadly across different kinds of cancer?
Yeah, so actually I first discovered this combination therapy in prostate cancer.
Okay.
So during my postdoctoral research up in WSU, we were studying prostate cancer, and we found that when they become resistant to chemo, they have high muscarinic receptor expression. Let's throw on an anti-muscarinic, and it wiped it out. And we thought maybe this also applies to other cancers. And so we got lung cancer cells, ones that were sensitive, ones that were resistant, and we did the experiment that I've already explained, and it worked. And so we expect that if we look in colon cancer, if we look in brain cancer, if we look in other different types of cancers, when they become resistant and they start expressing muscarinic receptors highly, you could use this combination therapy to treat. I know research has been done in breast cancer that has shown a similar thing. Outside of that, I'm not familiar who's done that, but it would be research we would love to expand into.
Yeah, because that could be a huge leap if you found it in one thing and it applies to all cancers. That'd be
Massive.
massive. Where do you see this study? What is the hope within the next call it 10 years.
Well, so we've published our results. What we did is we looked, this was in prostate, excuse me. We've published our prostate results. And this was in a dish, in a mouse model. And then we actually got patient samples, tumors, like actually solid chunks of tumor from patients that we were able to inject into mice and grow them. And so this is
interesting.
this mimics the patient much better than the cells we buy at the store.
Right.
And that all worked. And so the next step is clinical trials. And unfortunately, as I mentioned earlier, there's no money in this for drug companies. And so nobody's going to want to fund this. And so we're just doing what we can as scientists, is we think this is a really good therapy as a community, as a scientific community. Here's that information. And so we've published those results. And then I'm furthering that research into lung and then potentially moving on even farther into colon and so forth to just say, here it is. Whoever wants to, whatever philanthropist wants to fund this.
Advance the cure to cancer.
I wouldn't say cure. An effective treatment. It's available. This is an option. What's also nice is because these drugs are already used in patients, there is a potential for clinicians to see this research and go, this isn't a new drug. Like, I could already use this in a patient.
Yeah.
Let's try it. And particularly because the chemotherapy that we are resensitizing to, that's last line in prostate. That's last line in lung. And so maybe a patient will go, I have no other options. Let's try it.
That's generally when these things happen.
Yeah.
Is there a hope? Is the hope that you mentioned that chemotherapy is more of just a last step of like, we're just going to hold it off as long as we can? Is the hope that with these anti-muscarinic receptors drugs, we are able to kill the cancer at that final stage, or is it simply more effective in holding it off?
I don't know.
Okay.
So I can't say that it will entirely wipe it out or if it will just keep it at bay. Honestly, though, either option is better than it progressing.
Than it at the moment.
Correct.
Yeah. What can people do, the regular people, to help speed this process along?
What could the like community at large help speed this up?
Yeah.
We are in a very interesting political climate right now with scientific funding. And so any support, either writing your legislature, writing your government officials, anything like that is let's fund science. Let's fund researchers to help you to come up with treatments for you in the future. That's what I can see being helpful.
And good science. We want good science. I know that
Yes.
Trump has cut $50 million. I think maybe it was just $8 million to see if we could transgender the rats. So we're not trying.
I don't want to take a political stand.
We're not trying to. You're not trying to fund all science, but
I would like at
least
to fund my science.
Yes. Yeah. Talk to me about Medimon.
Yeah. So so what we've talked about so far. That's my bench research. I am a medical education professor, so I teach in the med school here in WWAMI. And that's kind of my other branch of research is education research. How can I better train future physicians in order to treat the community at large better? And the amount of information that medical students are required to learn is astronomical.
Nuts, yeah.
And it's only getting worse. And so what I'm trying to do is how can I come up with ways that they can better not only learn, but memorize and retain this information long term.
Right.
Not only for their board exams, but hopefully in the future when they're actually treating patients, they'll remember, oh, Dr. Bland taught me that one thing, and I can now cure this patient. I don't know.
Yeah.
And so one way that I am doing that is I've created an educational video game called Medimon. What this is, if anybody listening is familiar with Pokemon, Pokemon is a monster taming game where you travel around, you catch these creatures, you battle them, you grow them up. And Pokemon stands for pocket monsters because you keep them in your pocket. Medimon is very similar, but all of our monsters represent something in the body. The lungs, the heart, the kidneys, the muscles. and it's called Medimon, which is short for medical monsters because it's all medical. We even have diseased ones. And how it teaches students is that we have taken important characteristics of whatever that monster represents. So, for instance, if it's a cancer one, which we don't have yet, that'll be in Game 3. What we do is we take important characteristics of, say, it's expressing protein X. We put that in its design, in its visual design. It might have a big X on it. And so that way my med students, and maybe even students even lower who are interested in med school, high school students or so forth, they can just remember what that character looks like, and they can know, oh, I know that cancer expresses protein X because it has a big X on it.
Right.
And so these are called mnemonics. These are memory hooks in order to help people remember. So not only am I trying to help them remember, but hopefully they can have fun doing it. Because another big problem in medical students is burnout. Is like we are just force feeding them this information day after day after day. Maybe they can take a break and play this, but then I go, just kidding, you're not taking a break, you're still learning, but in an enjoyable format.
Yeah. Where have you seen efficacy, success with this way of learning?
Yeah, so we're currently running a study right now with University of Idaho undergrad students where we have a NASA grant in order to help teach undergrads important aspects of space medicine. And when astronauts go up into outer space is not very nice to them. And so they can get osteoporosis, they can get muscle wasting, they can get different eye disorders. And so in order to promote not only understanding of these important disorders and body systems, but maybe to promote transition into a career to help astronauts in the future, we've developed a demo that has characters that are related to those systems. So we have bone characters, we have muscle characters, and we have eye characters. And what we do is we allow undergraduate students to play the game. We test them in advance on their knowledge of it. We have a short survey that they take. What does different cells in the bones do? What do the different layers of the muscle? We measure their baseline knowledge of that, let them play it, and then test them again afterwards to see did they learn anything from the game.
Right.
And preliminary data shows that it increases their scores by about 50%.
Which is pretty good.
Which is pretty good, being as we are not force feeding them anything. We are going, here's the game, play it as much or as little, play it however you want. So it's self-directed learning. Hopefully they're enjoying it. But again, secretly I'm teaching them.
Yeah. Have you seen a limit thus far to the amount that can help?
Yes, of course. So we are not able to put everything that students need to know about these different organ systems, these different cell types into the characters. Correct. It would be an absolute mess. This was one thing we ran into when we were originally developing these characters, is I was developing what all should go into the character, and I worked with University of undergrad at the time students, Sierra and Emma. I'm calling you guys out. They were artists that made my vision a reality, that took my sketches and converted it into the beautiful artistic designs that you see today. And there was always a battle between us going, I want to fit more in this. And they would come back and being like, it can't look good. Like there's too much. It's too busy. We need to simplify. And so how we landed on was what are what are the biggest characteristics or things that students should know about the skeleton? And we put it in that. So we typically try to go with 2, 3, or 4 of the main characteristics to put in, and we try not to go farther than that.
Yeah.
So because of that, we purposely limited ourselves on you're not going to learn everything you need to know from this.
Yeah.
But hopefully you know the main parts.
But hopefully they can somewhat memory palace attach different items to that in their mind, and
correct.
That's the goal.
Correct.
Yeah. Yep. Is there a plan to roll it out to the rest of the University of Idaho or even beyond that?
Yeah, so viewers can play it right now. If you go to medimon.games, you can see all of the creatures we've made. You can download the demo. The demo's for free. It's our first level of the game that you can travel around, catch them, battle them. We don't say collect them all because that's Pokemon, but because we have disease states, we are contract them all.
There you go.
But yeah, so anybody can play it right now.
Yeah.
It is an ambitious game, and so every year we are going to release it next year. And so I'm I'm hoping next year, but in reality, next year will probably be next year.
There you go.
But as of right now, yeah, people can play the demo and give us any feedback that they
Yeah.
like it, they dislike it, this is broke.
Yeah.
hmm
Given our conversation today as we wrap up here, what's your best piece of advice you would give to anyone watching?
My best piece of advice, probably the same advice that I gave for how can the community help. Talk to your legislature, vote for people that you believe in, and I hope that those people believe in furthering science, at least good science, because I think it's important that the U.S. stay the leader in scientific research and medical research in particular. So help us out.
Yeah. Thank you so much for coming on the show.
Thanks for having me.
People can find medimon.games. Anywhere else they should look for you and your work?
You can find me on LinkedIn. I also have so I teach pharmacology at WWAMI. I have a pharmacology online course-esque at blandpharm.com.
Fantastic.
Yeah.
I'll put all the links in the description.
Thanks
Thank you so
for having
much
me.
for coming on the show. See you guys.